As Andy said, I've been working on annelids, and today I'm going to talk specifically about the role of gene duplications in the invasion of freshwater environments by metazoans, which are animals, annelids and mollusks specifically, but a few other groups as well. So the plan for today, give you a little bit of a introduction and background, kind of how I got here, talk about ATPase gene duplications and annelids, then talk about various gene family expansions in freshwater spiralian animals, and then finally talk about specifically the sodium-potassium ATPase gene duplication in freshwater mollusk lineages. And then I'll try to kind of wrap things up with a little general discussion. So first of all, as Andy mentioned, I came here as part of the Annelid Tree of Life project. We were trying to assemble kind of the family evolutionary relationships of annelids, which are segmented worms. And yes, that includes earthworms and leeches, what people usually think of when you talk about worms. But there's actually a lot of really other cool annelid worms, like these feather dusters, these Christmas tree worms, bristle worms, and everyone's favorite, the bobbit worm, which look it up on YouTube for some great videos. But as part of that project, we were producing very large amounts of data, sequence data. We were sequencing a lot of transcriptomes. We were assembling phylogenies, but I started trying to think about other things we could do with those data, other questions we could address. You know, what are some other interesting things we can look at in annelids? And annelids are actually a really cool group to study evolution in, because they are diverse. They have a lot of different lifestyles, a lot of different habitats. So the first thing I looked for is to see what genomes were publicly available for annelids. And what I found was there were two well-annotated annelid genomes out there, Capitella tallata and Helibdella robusta. These are cool. There's Capitella right there. This is a small annelid phylogeny, and there's Helibdella. And one of the things I thought right away was this is a really cool sample because it spans kind of the major marine freshwater divide in annelids, right? This yellow box here represents the clydolate annelids. That's a clade within annelids. Most non-clydolate annelids are marine. They live in the ocean. Most clydolate annelids are freshwater or terrestrial. And so if we look at this branch right here, any, kind of molecular adaptations that we expect to be heavily involved in the colonization of freshwater environments likely occurred somewhere along this branch, along this lineage. And if I show you that on another phylogeny, I don't know how well you can see the little circles here, but here is that same branch, the base of the clydolate tree. If you see outside of that, these are mostly marine organisms. And then inside of this is mostly freshwater, some terrestrial, some going back to marine, which is cool. And then a big group down here going into terrestrial environments. So this is a really cool question to study because it doesn't happen very often. If you look at the history of animal evolution and the major groups of animals, there's about 35 extant animal phyla, so with living representatives. Only about half of those have freshwater species. All of these phyla evolved in the ocean. Only about half of them have any lineages that manage to cross that salinity divide and make it into freshwater. And of those, the vast majority of freshwater species are in just 6 phyla. Arthropods, so that includes crustaceans like crayfish and insects. Chordata, that includes fish and mammals. Mollusca, the mollusks, snails and slugs, things like that. Annelida, which we're looking at, the worms. Nematoda, another group of different worms. And Rotifera, weird little rotifers that live in the water that I think are really cool. Most people don't think about at all. So the opportunity was there to start looking at this transition, this marine to freshwater transition. And if we think about this, we know this requires extensive modification to ionic regulation and osmoregulatory functions in the body. And doing a little bit of digging, one gene that is known to be involved in these salinity transfers is the sodium-potassium ATPase gene. So this gene pumps sodium ions out of the cell and potassium ions into the cell, okay? And it pumps those ions against the concentration gradient. So that does a few things. So first of all, this gene is part of a very old, well-conserved superfamily of genes known as P-type ATPases. They're all membrane-bound proteins. They all move stuff across the membrane. This particular one moves sodium and potassium across the membrane. It's involved in osmoregulation of the cell. So as you can imagine, that's going to be important if you change the habitat you're living in. But it's also really important in maintaining ionic gradients across the cell membrane. So your muscle cells, your nerve cells, they all function based on maintaining an ion gradient across the cell membrane, which creates a voltage across the cell membrane. This is the protein that does that, that maintains that. And we have seen, though, this gene is also upregulated during salinity transfers. If you take some animals that can survive in a range of salinities, you move them from ocean water to fresh water, you will see more activity transcribing this gene, more copies of this gene getting transcribed. So it was something of interest, something I wanted to look at. And a big question is, if this is so important in just maintaining regular function, how do we expect it to evolve for new habitats? So I decided to look for evidence of gene duplications. And the reason for that is gene duplications potentially allow for the evolution of highly conserved genes. You can, if you duplicate a gene, you can preserve one copy in its normal function while the other is free to evolve. It can also potentially allow changes to the regulation of the gene, a regulatory function. And the other thing is it can simply be used to increase the dosage of the protein that gets made from that gene. And we see that this gene is upregulated during salinity transfer. So I downloaded those available annelid genomes, the Helibdella and Capitella. I also grabbed a couple marine mollusk genomes just to increase the sample size a little bit. And then I used the PFAM database, which is a database of protein annotations to find all the P-type ATPases in those genomes. And then I did a maximum likelihood tree of all the P-type ATPases in those genomes, found all the subfamilies of that superfamily, and I identified 3 that showed a clear increase in size in the Helibdella lineage. So if we look at those are the plasma membrane calcium ATPase, the sarcoendoplasmic reticulum calcium ATPase, and then that gene I was just talking about, the sodium potassium ATPase. If you look at the sodium potassium ATPase here, Crassostrea and Ladia, those 2 marine mollusks, each have 1 copy. Capitella, that marine annelid, has 2 copies. Helibdella, the freshwater annelid, the leech, has 12 copies, which is a pretty striking difference. But it's just a sample size of 1. And Helibdella is kind of weird. You know, so are these duplications characteristic of freshwater annelids in general, or just Helibdella for some other reason, or just leeches, or just something else, right? So I only had 2 annelid genomes. I couldn't answer that question. But all those data we were talking about producing, I had a lot of transcriptomes. So as a brief review, DNA gets transcribed into RNA, which gets translated into protein sequences. If you sequence all the DNA in an organism, you get what we call the genome. If you grab all that mRNA and sequence that, you get what we call the transcriptome, which is a sequence of all the genes being actively transcribed. And if you sequence the proteins, you get the proteome. You can also infer the proteome from the genomic or transcriptomic sequences. And for the rest of this talk, when I talk about sequences, I'm going to be talking about the amino acid sequence of proteins that we inferred from the transcriptomes and genomes. So I had lots of transcriptomes, but problematically, for a number of reasons, transcriptomes are not good for counting the number of copies of genes in a genome, which is really what I want to do. But there's something else I can do with the transcriptomes. For any transcript that shows up in the transcriptomes, I can use that to make a gene tree. And if I have a species phylogeny and a gene phylogeny, okay, I can use the reconciliation between the two to infer the locations of gene duplications. So here is a speciation event, and you see the line in the center represents the lineage of a gene, and this 1 copy becomes 2 as it goes into, 1 into each lineage. So that's a speciation event. Here is a duplication event where within a single lineage, 1 copy becomes 2. So if I produce some species tree and some gene trees, I can infer the timing of these duplication events. And so the question is, where did these gene copies arise in the annelid phylogeny? I know they're present in the Helibdella, but do they coincide with that major radiation into fresh water? So my plan became use the transcriptomes to infer a species phylogeny with more than just 2 annelid species. Find the ATPase gene transcripts in the transcriptome, infer gene trees for each set of ATPase genes that I'm interested in, and then reconcile those gene trees with the species tree to determine the timing of those gene duplication events. Okay. So, briefly, as I talk about inferring species trees and gene trees, I just want to point out that to infer a species tree, what's most valuable to me are these gene copies that arose through speciation events. And if possible, I want to limit my data set to just those. To infer a gene tree, I want all the gene copies, the ones that arose through speciation events and the ones that arose through duplication events. So I need to use two different methods to identify those genes in order to infer those trees. So a basic workflow that I used is for each species that I had a transcriptome, all of these little boxes represent the different amino acid sequences. To infer my species tree, I used an orthology assessment method that would find one copy per species for each of the different genes. For my gene tree, I needed to use an orthology method that would just group every copy of each different gene together, keeping multiple copies per species if they were present there. So for the species tree, I get that. I then align each gene sequence, concatenate them into one data matrix, and infer my tree. For the gene trees, similar process. I have my orthology groups. I align the gene sequences or the amino acid sequences at this point for each gene, each of the different genes, and then infer a tree from each one. So this is the tree for this gene, then infer a tree for this gene, which I don't show. And these gene trees can have multiple genes per species. Mostly for the species tree, I use a program called Hamster for this, and use a program called RaxML to infer the species tree. To infer the gene trees, I had to do something a little more complicated. So first of all, with the species tree, I don't really care what the genes are, as long as I have single copy orthologs. For the gene trees, I needed to know which ones I wanted to use. So I used the Interpro database for annotations for the sequences to figure out what each gene was, to find my p-type ATPases to make those gene trees. And then to use to infer the gene trees, I also use a program called TreeFix because for a number of reasons, you can get a lot of uncertainty in your gene trees, just because of shorter sequences, more similar sequences, things like that. And you can end up with a lot of different topologies that are statistically equivalent. And so this program will take the statistically equivalent ones and find the ones with the least number of duplication events. And so if I'm erring, I'm erring on the more conservative side. And then I use a program called NoTongue to map the duplications to the species tree. Okay. So here is the annelid phylogeny. I produce the species phylogeny I produce. This is the group of clitollates I used. Here is kind of the base of the clitollate tree where I'm expecting the gene duplications to appear if they contributed to that invasion of fresh water. And then here is indicating how many duplications I inferred on each of these branches. And you can see most of them occurred right at that base of the clitollate tree where I would expect them if they were involved in that transition to fresh water. The two different colors here represent two different data sets I had. I found I had a lot of truncated sequences, shorter than I expected. So I did one data set that was just the complete transcripts and one that was just a minimum of 300 amino acids long. And either way, whether I use just the complete transcripts or I use both sets, you see the same pattern. You're seeing most of these duplications are arising right there at the base of the clitolate tree where that colonization of freshwater occurred. And then, thanks to another project we had going through the Annelid Tree of Life project, this chronogram where we try to time some of these divisions, we can actually put a real time on that. So this is that base of the clitolate tree I was talking about, where we see most of those duplications occurring. And we now know, thanks to our group, that occurred kind of between 450 and around 400 million years ago. So we can put a real time to it. So part one, conclusions. There was an increased number of copies of the sodium potassium ATPase gene in a freshwater annelid compared to a marine annelid. And when we inferred the timing of those duplication events, we found they occurred at the time that corresponds to this major invasion and radiation into freshwater by annelids. Okay. For the next thing I did, I wanted to kind of expand my search to new gene families and open it up to kind of new species as well. So I needed more genomes. And so I expanded it to a group called Spiralia, which includes annelids, mollusks, and several other phyla. Here is the phylogeny of the species I use. The asterisk here represents a freshwater species. So I have platyhelminths, a rotifer, both freshwater, one marine annelid, one freshwater annelid, a marine brachiopod, some marine mollusks, one freshwater mollusk. Ended up with 7 marine animals, 4 freshwater. And then I use a similar process to the species tree inference to get that species tree. Although some of the analyses I did with this one, branch lengths matter. So I did an extra filtering step to filter out some of the groups that might disproportionately affect the branch length. And I also needed an ultrametric tree, even tip points. So I used the Chronos function in R to do that. Amino acid sequences for each of the genomes were assigned to orthogroups using OrthoMCL. This is to get the gene family sizes for each. And then I used a program called CAFE to determine which of those orthogroups, which of those gene families showed a significant change in size. So CAFE will kind of map the changes in gene family size to the phylogeny and show you which branches on the phylogeny underwent a significant change in size. All right, so we need to talk about gene ontology. So you can think of an ontology as kind of a concept map of everything we know about something. In this case, biological function or molecular function of proteins. And then every protein gets assigned whatever Go terms are relevant to it. And some will be kind of parent-child relationships. So if you think about the sodium-potassium pump, it will be assigned the Go term transmembrane protein. And then under that, transmembrane ion transport. And under that, transmembrane ion pump, cation pump under that, right? And what I'm using the Go terms for is to take all of the gene families that underwent a significant expansion and see which functions are overrepresented among that group of families. So you can take all of the gene families with all of the Go terms that are assigned to those as kind of your universe, and then take just the ones that experienced expansions in the freshwater lineages as your subset, and see in that subset what functions are overrepresented compared to what you would expect if you were just randomly picking a subset. And so what I found was, oops, let me go back. I'm sorry about that. Oh, my button's stuck. Sorry. So what I found was that out of the entire gene ontology, a couple very small subgraphs that showed significant results. This is biological process. This is molecular function. We look at biological process, you'll see things like regulation of transport, regulation of transmembrane transport, regulation of ion transport, and this is what I was saying when you get those parent-child relationships. Regulation of ion transmembrane transport. So this is the part of the ontology that kind of lit up, and the pink boxes represent the results that were significant. And then you look at molecular functions, kind of same type of thing, voltage-gated channel activity, voltage-gated ion channel activity, voltage-gated cation channel activity, ion transporter membrane activity. So overall, function-wise, we're seeing transmembrane ion transport seems to be the overwhelming function that we see in these gene families that underwent expansions in freshwater lineages. So then I wanted to ask, okay, those are the functions, whether it's the same specific gene family or not. Now, are there specific individual gene families that seem to have gone through this repeatedly and independently on these freshwater lineages? And for that, I use phylogenetic ANOVAs of the gene counts in marine versus freshwater. So a phylogenetic ANOVA simply does an ANOVA, but ANOVAs assume samples are independent. If you're dealing with organisms, they're not independent, they're related through their phylogeny. So a phylogenetic ANOVA accounts for the phylogenetic effects when comparing, groups that are related like that. And what I found with the phylogenetic ANOVAs, so these are the names of the orthogroups. I found about a dozen that showed a significant hit even after correction for false discovery rate in there. And, we look at some of these domains that are in these protein groups: ion transport channel, calcium transport, ion transport protein, E1, E2 ATPase. So that E1, E2 ATPase is another name for the P-type ATPases. E1, E2 just refers to a conformation change. This one, OG51270003, that is the sodium potassium ATPase I've been talking so much about. And then just to show these weren't caused by a few outliers, I did box plots for all of them. And you can see on these box plots, a lot of these don't even overlap between the freshwater and the marine. You have a real striking difference. And again, if we highlight that sodium potassium pump right there and take a closer look at it, you can see basically none of the marine animals seem to have more than 3 or 4 copies. None of the freshwater have less than, looks about 7 copies. And some of the freshwater species have up into the 30s. So really, really dramatic difference. So we can see on each freshwater lineage, this family underwent independent expansions. So part 2, conclusions. Gene families related to ion transport underwent significant expansions in freshwater lineages, independently and repeatedly in freshwater lineages. So for part 3, I decided to really zero in on the sodium potassium pump, but really because I wanted to ask a specific question. In part 1, I looked at annelids, and there's that one major lineage leading into the freshwater radiation. In mollusks, you have a lot of freshwater radiations. So mollusks are a large and diverse clade. They have lots of marine species, lots of freshwater species, lots of terrestrial species. If we look at the major groups in mollusks, you know, bivalves and gastropods. So bivalves are things like clams and mussels, gastropods, slugs, and snails. Each have multiple marine to freshwater transitions. You know, in gastropods, last estimate I saw was at least 35 or so. And they have lots and lots of freshwater species. So you look at other mollusk groups like cephalopods, they haven't colonized freshwater at all. Something is seemingly preventing them, but gastropods seem to be able to do it no problem, over and over again. So my question, number 1, do we see these same sodium potassium gene duplications in freshwater mollusk lineages? And if so, when did they arise? Do they arise independently corresponding to each colonization event? Or is this something that arose earlier in the phylogeny? And is that why this group now has been so successful in being able to do this over and over again? So first thing I did, I downloaded the protein sets from 14 mollusk genomes. Then I downloaded a bunch of RNA-seq data to assemble transcriptomes so I could get more species in the sampling. Out of all the transcriptomes I assembled, only 21 met our completeness threshold. So problematically, a lot of the people who produce transcriptomes are not doing it to do this type of work. They're interested in what genes are being up or down regulated in a particular tissue at a particular time. So they end up just sampling particular tissues, and you don't get kind of a good picture of all the genes in the organism. So a lot of these, when we looked at the expected number of core genes, we weren't getting anywhere close to completeness. I think I lowered my completeness threshold to 40% just so I could, you know, get up to 21 of them. But that did give me 35 total taxa spanning several marine to freshwater transitions. So species tree was inferred similar to how I said it before. This time I used Busco instead of hamster. I did a species phylogeny using only the genomic sequences, the 14 taxa. And I did that for use in the Phylogenetic ANOVAs that I performed. And I did a species phylogeny using both the genomic and transcriptomic taxa, the 35 taxa species tree. And I used that for the species tree, gene tree reconciliation. For the gene tree inference, same basic process, used OrthoMCO to assign sequences to orthogroups, aligned them with MAFT, and then I used a program called GeneRax to infer the gene trees, and it's a species tree aware gene tree program. So it minimizes the number of duplication events as it's inferring that gene tree. So again, if I err, it's going to be on the more conservative side, hopefully. And then again, use no tongue to reconcile the gene tree and the species tree. So what I found first with the phylogenetic ANOVAs, so down here in bold are all of the freshwater species. And I tested the 12 orthogroups from the spiralian analysis that showed a significant expansion in freshwater lineages. In this case, the only one that appeared significant was the sodium-potassium ATPase group. So part of this is limited sampling in both of these studies, right? It's going to make for part of the discrepancy. The other thing, notice all of my freshwater species here are clustered, and the phylogenetic ANOVAs kind of take that into account. So it's going to have to be a much, much stronger signal to be determined significant than if I had freshwater species kind of spread throughout the phylogeny and saw the same number differences. But either way, this is something I'd like to revisit in the future. But point being that sodium-potassium group is popping out in every study, no matter what I do. And then the other thing, the gene duplications. So there's some question over where scaphopods belong. This is the species tree I inferred, and it put the scaphopods outside the bivalve gastropod split, which is where a lot of people put it. Recently, some have suggested scaphopods go as sister to gastropods and bivalves outside of that. So I did a constraint tree to force that to happen so I could do the analyses both ways. And then what I found when I mapped the gene duplications onto the species phylogeny, is I see a number of gene duplications, more than any other branch, occurring prior to that scaphopod bivalve gastropod split. And if I put scaphopods inside there, I see it again occurring prior to the split, and then another big round of duplications prior to the scaphopod gastropod split. This was a bit of a surprise to me. I figured if the scaphopods were outside of the bivalves and gastropods, most of the duplications would occur after the scaphopods split off, because I didn't expect there to be a bunch of these gene copies in scaphopods, because as far as I know, scaphopods are pretty much restricted to marine environments. Bivalves and gastropods both have a lot of freshwater species. And again, in bold here, you can see my freshwater bivalves. You can see some additional duplications there, but I'm not going to talk too much about bivalves because I had very limited sampling on that. Within the gastropods, the Vetagastropoda is marine, the Patelogastropoda is marine, and then the Neridomorpha, Heterobranchia, and Cenogastropoda all have a lot of freshwater species and all have several independent freshwater invasion events. So kind of, again, between the three of these, you have at least, 35 independent freshwater invasion events. I don't have real good sampling within each of these groups, but I do have representatives from all of them. I have representatives from the marine groups outside of them, and I have for most of these, well, for the Heterobranchia and Cenogastropoda, I have at least some marine species and some freshwater species. So what we see, this big round of duplications prior to the bivalve gastropod scaphopod split. And then within the gastropods, where you have clades that have become really successful at invading fresh water, you have more rounds of duplications leading to these clades. And again, this is the sodium-potassium duplications. I don't know about any duplications on the branch leading to Neridomorpha because I only have one species from that. And basically, because of transcriptome issues that we can talk about as long as you want later, I basically ignored all the species-specific duplications that were inferred. So I had to ignore all the duplications on that branch. So this would be nice to do again with more sampling from there. But the conclusions from this, the sodium-potassium gene duplications are present on branches leading to clades with freshwater invasion events. There are no sodium-potassium ATPase gene duplications on branches with no freshwater descendants. The duplications seen prior to the split of bivalves and gastropods possibly contributed to their future success in colonizing fresh water. Again, since I'm getting these data from transcriptomes, we know these genes are still being actively transcribed and used in some manner. So, and then that does leave the question, what about Scaphopoda though? I don't know. I had a really nice, neat, beautiful story to tell. If it weren't for those meddling scaphopods, I don't know what they're doing. I feel like I'm going to regret not knowing what they're doing in the next hour with my committee, but we'll see how that goes. So, conclusions, additional duplications on the branches leading to Heterobranchia and Cenogastropoda, which are both groups that contain many transition events. Overall, more and better sampling is needed, definitely for bivalves, but also for those three gastropod groups, the Cenogastropoda, Heterobranchia, and Neridomorpha. It'd be great to do more sampling. If anyone wants to pay for some collection and genome sequencing, let me know. So overall, general discussion, what does all of this mean? I don't know. Sodium-potassium ATPase gene duplications seem to play a role in the successful colonization of freshwater habitats, at least by annelids and mollusks, likely other spiralian animals, and honestly, likely other animals as well. You know, we could expand this out, start looking at arthropods and stuff as well. Most of the work on arthropods is done just kind of real time by like saline tolerant animals, and they're just looking at like what genes are being expressed in real time. I don't think a lot of is being done on the kind of evolutionary time scale. Freshwater species continue to express at least some duplicate copies of the sodium potassium ATPase gene. How exactly are these extra copies of the sodium potassium ATPase gene contributing to adaptation to freshwater? I don't know. What about the beta subunit of the sodium potassium pump? So here's a little secret. The sodium potassium ATPase is a multi-domain protein. That means several amino acid sequences coded by different genes come together like Voltron to form the fully functional protein. I have only been talking about the alpha subunit, which is the transmembrane part. There's also a beta subunit that plays a role in kind of positioning the protein as a whole into, you know, what tissue it's in, what membrane it's in, things like that. And I feel like there must be something going on with that as well. Finally, some transcripts were much shorter than expected. Is this just sequencing error? That's probably too optimistic. Pseudogenes continuing to be transcribed because, again, they are being actively transcribed. And if they're pseudogenes, why? And why just in the freshwater lineages? Or is there something else going on? That'll be worth looking into. So overall, takeaways, more and better sampling is needed. Gene expression studies are needed. I would really like to see kind of if we could nail down individual copies of this gene and what they're doing at different times. And then also what other genes are involved. So I started trying to look at that with the spiralian genomes. I limited my sampling of genomes for that for a reason that I could get around now. So I'd actually kind of like to go back and revisit that with better sampling. Just as a starting point of like, oh, look at these other groups as well. But yeah, there's, you know, I think all of this is really, really interesting. And I think it kind of creates more questions than answers though. So there's a lot of directions people could go with this. So acknowledgements, I'd like to thank my advisor, Dr. Anderson, who has, you know, put up with a lot for me. My other committee members, Augustine Jimenez, Dan Nickren, Richard Thomas, and Ken Halanich. I'd like to acknowledge the National Science Foundation for the funding we got through the WormNet 2 Assembling the Tree of Life annelid grant. Thanks to SIU and the Department of Zoology, my friends and family members, and a special shout out to the cellar for being a home and hideout and sometimes sanctuary for me. With that, I'll leave it up to questions. This is a picture of a little leech I'm currently keeping as an office pet, trying and failing to attack an earthworm. So we'll just leave this with a little annelid on annelid violence and open it up for questions.